Good morning, everyone. So my name is Arianna, and this summer I had the pleasure of working at the Nijagal lab at UCSF, looking at the impact of macrophages on hepatoblastoma with Dr. Amar and Claire. And so today I'll be sharing a short summary of what I have worked on. So it is no doubt that cancer is a devastating diagnosis. Hepatoblastoma is rare and is usually found in babies and children up to age 3 and is the most common liver cancer of childhood, although it affects only 2 to 3 people in a million. Immunotherapy is regarded as the most promising treatment for cancers because the immune system has been known to be critical in fighting cancer. And interestingly, macrophages, which is a type of immune cell, has been identified in the tumors of children with liver cancer. And by secreting various factors and affecting other immune cells, macrophages not only play a role in chronic inflammation, but also initiate, promote, or suppress the development of cancer. And as you can see in this pictures, macrophages have been demonstrated to be involved directly or indirectly in several key features of malignant tumors, including angiogenesis, invasiveness, metastasis, regulation of the tumor microenvironment, and therapeutic resistance. However, there's currently little to no research examining the role of macrophages on liver cancer. Therefore, better understanding of the mechanisms underlying the function of tumor-associated macrophages is necessary for the development of novel tumor-associated macrophage targeting immunological interventions, which may provide promising therapeutic approaches for liver cancer patients. Because remember, this type of liver cancer affects mainly children, which makes it all the more devastating. So overall, my project aimed to evaluate and address the research question of what is the role of macrophages on hepatoblastoma tumor cells. And to help answer this research question, I first analyzed existing data from my lab's database to look at macrophage gene expression in liver cancer. Here on the left, you can see a list of genes, and highlighted in red are genes that are overexpressed in liver cancer. And note that the categories of the gene may overlap. So these results assert that tumor-associated macrophages promote tumorigenesis by expressing certain genes. And the picture on the right offers a schematic representation of the impact of the tumor-associated macrophages in promoting tumor progression. So for instance, the overexpression of VEGF promotes angiogenesis and tumor growth. The overexpression of CSF1 and MMP secreted by tumor-associated macrophages in liver cancer promotes tumor migration and invasion. And to help further build upon the ideas that macrophages promote pro-tumor activities in liver cancer, Claire performed an experiment. So using an in vitro co-culture system, an EDU assay was performed to help measure the growth of tumor cells in liver cancer. And so on the left side of my screen, you can see the well plates that were seeded with liver cancer cells, macrophages, and monocytes. And the EDU assay measures the cell's ability to proliferate using flow cytometry or fluorescence microscopy. And this helps assess cell health, determine genotoxicity, and evaluate anti-tumor activity. And then on the right of my screen, you can see how macrophages promote liver cancer because the liver cancer is faster growing and more aggressive. So these results again assert that macrophages are key drivers of cancer progression. And with this co-culture assay, exploring and identifying what genes and macrophages affect tumor growth and liver progression would be the next steps in hopes to categorize pro-tumor and anti-tumor associated genes. However, macrophages may not be completely bad after all. The powerful two-edged nature of macrophages offers a unique opportunity. And like I mentioned earlier, liver cancer in children is a problem, but it is not studied enough. So results from my time at the Nijagal lab allows for the discovery and better understanding of characteristics and interactions between liver cancer cells and macrophages, which helps reveal what genes are good targets for anti-cancer therapy in humans. So for example, one of the many genes overexpressed in liver cancer was CCR2. And so CCR2 promotes immune suppression, and high expression increases tumor growth, induces cancer cell migration and survival, and results in poor prognosis. So inhibiting CCR2 could potentially decrease the tumor burden and progression in liver cancer. And furthermore, targeting other genes listed in the left-hand box here could alter the functions of macrophages as well. And then using Claire's co-culture assay, this can detect the impact of targeting genes and macrophages. So ultimately, it is extremely needed and vital to continue to research and bring attention to a macrophage's ability to reduce, reprogram, or disinhibit tumor-associated macrophages so it could allow for the development of novel and more effective macrophage targeting liver cancer therapies. And with that, I want to thank everyone for listening, and thank you to everyone at the Nijagal Lab and the PUMAS program. I really do appreciate this incredible opportunity I've had this summer.
Thank you, Arianna. Any quick questions? There is a question for you. What was the source of the macrophages and the monocytes in your experiments?
So for the monocytes, we used THP1 cells, and we also added PMA, which induced the differentiation from like monocyte to macrophage. And for like the liver cancer cells, we used human liver cancer cells.
Great. Thank you. Thanks, Arianna.